epithelial barriers. The first layer is made by epithelial cells, epithelial layer by themselves. We have a layer or some layers of epithelial cells that they are the first layer that are exposed to the pathogens or microorganisms or foreign substances. Okay? And this protection is made by physical, chemical, and cellular components. I will talk about them. The second layer is the connective tissue that is underneath of these epithelial cells. So underneath of the epithelial cells, we have connective tissue. And in connective tissue, we have various immune cells, a variety of immune cells over there. And the second layer is draining lymph nodes or lymphoid tissues. Near the barriers, we have lymph nodes or lymphoid tissues. They provide the third layer of immune protection and tolerance. What does that mean? It means that the first responsibility, the first layer of responsibility is by epithelial cells. So epithelial cells, please, epithelial cells try to protect by themselves as much as possible with different mechanisms. And mostly, for example, my skin prevent the entrance of microorganisms. It's made by the epithelial cells, or with different mechanisms that I'll talk about now. If the first layer cannot control the entrance, cannot inhibit the entrance, we will have some microorganisms in the underlying connective tissue. So this connective tissue, and in connective tissue we have many immune cells. So here we have involvement of more immune cells and a stronger immune response. The immune response here is stronger than the previous layer. So if the connective tissue components cannot control, we have another layer of immune response, which is involvement of lymphoid, secondary lymphoid organs nearby the barriers. So when we go from first layer to the third layer, the immune response is becoming stronger and stronger. The first layer is the weaker, but mostly successful. In the most of the time it is successful. But sometimes when it is not successful, we have involvement of the connective tissue. And if the connective tissue successfully control the infection, we won't have involvement of draining lymph nodes. And if both first and second layer cannot control, we will have involvement of draining lymph node and, of course, a stronger immune response. Recruitment of more cells, immune cells, to the site of infection. So there are the different epithelial barriers of the body. You've seen this figure several times, that the first layer of immune response is the barriers of the body. And the barriers of the body is not same. There is similarity between them, but they are not same in different barriers. So the type of the epithelial cells and immune cells is a little different between different barriers. For example, in the skin, this one, we have several layers of epithelial cells. Or in the GI, we have a thick and thin mucosal layer. So there is a difference between different in the both, structures of epithelial cells, the type of epithelial cells, and of course the component of immune cells is different in various barriers. So we are talking about the first layer of immunity system now. So we have, for example, here in oral epithelium we have just one layer of epithelial cells. I won't go into the details of all these organs. I will just talk about GI and skin today. Let me see. So first of all, I will talk about intestine immunity. Not all the GI, just intestine of the GI. We do not have time to talk about all parts of GI and then all other barriers. just intestinal immune system. It's important to know that all epithelial cell types express innate immune receptors. What is the general name for innate immune receptors? PRRs. Is a general name for the, so epithelial cells, it's logical. The epithelial cells are the first cells that are encountered with microorganisms. So they are the first cells that have to recognize the antigen, recognize the microorganisms. So all of them, different types of PRRs have epithelial cells have PRRs. So we can say that epithelial cells are a part of innate immunity. They are involved in immune response. We have some specialized epithelial cells. For example, goblet cells are a type of epithelial cells that produce mucus. Or we have another epithelial cell which is called Paneth cells. They produce antimicrobial peptides, which in short, they are named AMP, antimicrobial peptides. They are mainly produced by Paneth cells. Neutrophils have AMP in their granule as well, or some eosinophils. Antimicrobial peptides are something like antibiotics. Sometimes they are called natural antibiotics. That they kill we have different kind of antimicrobial peptides that they kill different microorganisms with different mechanisms. So we have several types of antimicrobial peptides that we do not have enough time to talk about all of them one by one. They are mainly produced in the intestine or GI by Paneth cells or other epithelial barriers. And mucus is produced by goblet cells. There is another specialized epithelial cell, which is called M cells. They are involved in antigen sampling of the intestine. That I'll talk about what antigen sampling means in the intestine. This is a schematic figure of different layers of immune response in the intestine. Okay, please. I think I'm talking to myself. Please have a look here. Most of you are not in the class, and least of you is in the class, and you do not listen. And it's very difficult for me to explain for a small group of people that do not listen. Ya Allah. You've had 30 minutes rest time. Okay, this one is several questions I can give from this figure. You are going to memorize almost all of the components of this figure. Almost all of the components of this figure has to be memorized by you. Okay, so have a look here to talk about them one by one. It's very simple, but you need to study. So it's a schematic figure of intestine, epithelial layer. I told you that there is 3 layers: epithelial layer, connective tissue, which in the intestine is called lamina propria, is the connective tissue, is a name for connective tissue of intestine, and draining lymph nodes, mesenteric lymph nodes or draining lymph nodes of intestine. So there are 3, and here is an epithelial layer. So there are the 3 layers of protection in the intestine that I will talk about them one by one. First of all, I will begin with epithelial cells. However, here, first of all, we have between epithelial cells some types of junctions which are called tight junctions. Okay, tight junctions. Tight junctions. And those tight junctions those tight junctions make a physical barrier for microorganisms, prevent the entrance of microorganisms. Here is the lumen of the intestine, and there are so many commensal bacteria or maybe pathogenic bacteria and food antigens over there. So these epithelial cells, there are between these cells, there are tight junctions that bind those cells together firmly and prevent the entrance of microorganisms. That's the first role of epithelial cells, making a physical barrier, preventing the entrance or passing of the bacteria to the connective tissue or inside of the body. And in some Paneth cells, some epithelial cells, including, for example, here, Paneth cells, they are specialized epithelial cells that they produce antimicrobial peptide, AMPs, and you can see them here. And they release the antimicrobial peptide inside the intestine. They are produced by Paneth cells. Or there are another cells, which is called goblet cells. They produce mucus. This mucus is produced by goblet cells. And there is an M cell, its role is antigen sampling, that I will talk about. So, what is the role of mucus layer on the epithelial cell? There is a mucus layer here, you can see it here. It's the mucus on the epithelial cells. The epithelial cells are masked, there is a layer of mucus over there. What's the role of mucus? That's right. They keep the pathogens, prevent the pathogens to get close to epithelial cells. Indeed, it's a type of physical barrier. Mucus is a thick glycoprotein, a very thick glycoprotein on the epithelial cells. I think. Okay, have a look here. There are epithelial cells, and this part is mucus. And first of all, mucus prevents the entrance or prevent bacteria to close to the epithelial cells, keep the bacteria away from the epithelial cells. Besides, in the mucus there are antimicrobial peptides. If some bacteria come into the mucus, those antimicrobial peptides will kill the bacteria. Besides, in the mucus we have IgA. You might have remembered that IgA is very important in mucosal surfaces. So we have IgA in the mucus, inside the mucus, on the epithelial cells. So what do you think about the function of IgA here? We have mucus, and inside the mucus there are IgAs. What's the function of IgA here? Yes, the role is protection. How IgA does this protection?
By neutralization?
Yes, by neutralization. The first role of IgA is neutralization. Those IgAs bind to bacteria and prevent the binding of bacteria to the target, to the epithelial cells. And by binding of several IgA to the mucus, a large complexes are made here, and those complexes are flushed out by the content of the intestine. You know that we have constant movement of the content of the intestine toward the rectum in the intestine. So they are regularly washing out from the surface of epithelial cells. So IgA, first of all, neutralize the bacteria and make large complexes of bacteria and antibody, that those complexes are washed out from the surface of epithelial cells. So we can say that mucus have at least three functions. First of all, it's a physical barrier. It's a thick glycoprotein that prevents the bacteria to get close to the epithelial cells. The second function is having antimicrobial peptide inside. And the third function is having IgA. They are the roles of mucus. All of them are inside the mucus. Have a look here. They are epithelial cells. They are epithelial. It's a real figure. It's a real figure. It's not a schematic figure. And they are microorganisms. Here, the green colors. The green color is shown like the microorganisms over there, and the blue color shows the epithelial cells. And this part is mucus layer, and you know that there is a space, a free space between microorganisms and epithelial cells. So this mucus makes a physical barrier between microorganisms and epithelial cells, prevents the microorganisms to get close to the epithelial cells. And there are some antimicrobial peptides here and IgA as well. And again here, you can see a free space here. And this part is mucus. So, what is the function of mucus? Summarize to me. Yes?
Mucus prevent pathogens to enter into the epithelial cells.
Yes, to get close to the epithelial cells. Make a free space between microorganisms and epithelial cells. Yes. What else?
Inside the mucus there is antibody, IgA.
IgA.
That neutralizes the
bacteria.
The bacteria
Thank
to
you.
enter the epithelial cells.
And antimicrobial peptides.
Yeah, pathogens. Basically, all of the pathogens.
No. There are antimicrobial peptides in this layer that they kill the bacteria. Antimicrobial peptides. Inside the mucus, besides IgA, there are antimicrobial peptides that they kill the bacteria that, for example, trying to get close to the epithelial cells. So, I talked about epithelial cell. Between epithelial cells, there are some lymphocytes. You can see them here, which they are called intraepithelial lymphocytes. The lymphocytes that are located between epithelial cells. Intraepithelial lymphocytes. They are mainly gamma delta T cells. Do you remember? We had two types of T cells. Alpha, beta T cells, that they are majority of T cells throughout the body. But we had another, a minority of T cells that they had gamma delta receptors. They are called gamma delta T cells. They are mainly in mucosal surfaces. So we have gamma delta cells mainly here, and they can kill the cells that are infected with microorganisms. What does that? It means, first of all, this mucus and its content prevent the microbiome to get close to the epithelial cells. But if a microbe successfully enters the epithelial cells, these lymphocytes will recognize that epithelial cells and instantly will kill that. Instantly, immediately. So by killing the cells that is infected with the microbe, so the source of the microbe, for example this one, is destroyed and prevent the proliferation of the microbe inside the cell and entrance of the microbe to the underneath of the epithelial cells. Okay? So until now, we talked about tight junctions of epithelial cells, physical barriers. We talked about antimicrobial peptides here. We talked about mucus, and we talked about intraepithelial lymphocytes. All of them are parts of the first layer, epithelial layer. The second layer is a connective tissue, lamina propria, which is underneath of the epithelial cells. And we have so many immune cells which spread in all parts of connective tissue. If the antigen cannot be controlled by the first layer, indeed, if the antigen cannot be controlled by mucus, by epithelial tight junctions, by intraepithelial lymphocytes, and if we have bacteria over here, we have some immune cells, including B cells, that they can recognize the antigen and they will produce antibody. B cells, after recognition of antibody, they will produce IgA, and IgA will transfer to the lumen. Or we have mast cells, that those mast cells will produce cytokine and recruit other cells. We have macrophages that do phagocytosis. We have neutrophils, we have dendritic cells and other immune cells. So we have a lot of immune cells in connective tissue that you know their functions. With different functions, they try to control the microorganism, prevent the entrance of microorganisms to the rest of the tissue of the body. Exactly under the epithelial cell, there is a compartment which is called Peyer's patch. Peyer's patch is indeed a structure that inside the Peyer's patch we have follicle. What is a follicle? Oh my God. Ya Allah. Follicle.
No, it's a group of cells.
Which type of cells?
Limited zone, base of the B cell.
B cells. Follicles are a structure that B cells are. No, mainly B cells. Follicle is for B cells. It's a B cell compartment. We have a dense population of the B cells inside the follicle. And we had primary follicle and secondary follicle. Primary follicle was a follicle that haven't been exposed to antigen, but secondary follicle was the follicles that after proliferation of the B cells, we call the follicle as secondary follicle. So inside the Peyer patch, we have follicles, and inside the follicles, we have mainly B cells. And out of the follicles, we have T cells, and dendritic cells, and macrophages. They are very similar, they are a little similar to lymph nodes. Follicles, T cells, dendritic cells, macrophages. Okay? Clear or not? There is no other way.
Yeah.
You have to memorize them. There is no other way. So, we have follicle here. On the follicle, we have M cell. The role of M cell is antigen sampling. What does that mean? It means M cell transfer the antigens from the lumen to the follicle. Transfer the microbes from the lumen to the follicles. To present the microorganisms to the cells that are inside the follicle. It means we have a group of cells here, adaptive immune cells and innate immune cells, including B cells, T cells, macrophages, dendritic cells, that those cells do not know anything about the lumen. in order to be able to know about the content of the lumen, we have M cells that samples the antigen and deliver to the cells inside here. If there is a pathogenic bacteria, these cells will induce immune response. But if the antigen is not pathogenic, we won't have any inflammation. Clear or not? There is a layer of epithelium, okay? And under the epithelium there is a pack of immune cells. And those immune cells do not know or not aware about the lumen, if there is pathogenic bacteria or not, or if the bacteria over here are pathogenic or normal flora. So they need a way to get away from the content of the lumen. M cells will sample the antigen and transfer and deliver the antigens without killing them. They are not phagocytic cells. They just transfer the antigen from the lumen to the Peyer's patch. Just Peyer's patch. They are just above the Peyer's patch. So deliver the antigens from the lumen to the Peyer's patch. And now the cells inside the Peyer's patch will check the antigen. If it is the pathogenic, it will induce inflammation. If it's not pathogenic, there is we won't have inflammation. Clear or not? And we have some dendritic cells exactly under the underneath of the epithelial cells, and they have some projects which they are like hands. They send their hands to the lumen and again take some antigens and check the antigens. So it's another way for sampling of the antigens of the lumen by dendritic cells. So dendritic cells send its hand to the lumen of the intestine and take the antigens as an APC and check the antigen and decode the antigen. If the antigen is pathogenic, will induce immune response, inflammation. But if the antigen is not pathogenic, it won't induce inflammation or immune response. So until now, I've told you two ways of antigenic sampling of lumen by immune cell. The first one was made by M cell, the second one was by dendritic cells. Why there is need for antigen sampling? There are two ways of antigen sampling. Why do we need antigen samplings by immune cells from the lumen of intestine? Yeah?
To check this antigen is immunogenic or not.
Pathogenic or not.
Okay.
To check the nature of the microorganism or antigens. If there is need for immune response or not. It's very simple. It's just a sampling and checking. So in normal circumstances, the main role of those compartments until now is just checking, sampling and checking. Samples and check, samples and check. If there is no pathogen, there is no immune response. But if there is pathogen, we will have immune response. It is important to know that in normal circumstances, most of the cells that are here are indeed the tolerogenic cells. What does that mean? For example, we have macrophages here, that those macrophages mainly in normal circumstances are M2 macrophages. T cells are regulatory T cells. So in normal circumstances, we have M2 regulatory, and for example, B cells produce IgA. Again, it's a regulatory antibody indeed. They cannot activate complement and inflammation. Why in normal circumstances we need regulatory, a high number of regulatory cells in connective tissue or in barriers? We need suppression. We do not need immune activation, because here is full of harmless foreign antigens, including food antigens and normal microbiota. So most of the cells located here are regulatory cells, and the rest of the cells, and even these dendritic cells, in normal circumstances, they are tolerogenic dendritic cells. tolerogenic dendritic cells do not have co-stimulators, and they induce tolerance. Do you remember that APCs without co-stimulator induces tolerance? So most of the dendritic cells here are tolerogenic dendritic cells. They do not have co-stimulators. The rest of the cells are activated effector cells. In contrast to other tissues. In other tissues, we have mainly, or in other secondary lymphoid organs, we have mainly naive T cells. But here we have effector or regulatory. What does this mean? The cells in the connective tissue or barriers are mainly regulatory or effector, not naive and activatory. Effector, some of them are effector, some of them are regulatory. Why do we need for these two group of cells in the intestine or other barriers as well?
Rapid immune response? Rapid immune response?
Yes, very important. We have effectors because we need rapid immune response whenever we have a pathogenic bacteria. And we have regulatory cells because we do not need immune response when the antigen is not pathogenic. So there is a balance between effector and regulatory, but effectors are ready to respond. Most of the cells here are activatory and effectory cells, or activated effectory cells. They are ready. If there was naive cells here, activation and induction of immune response by naive cells takes time. It takes, for example, 1 week time, and it's very, very late for immune response in the intestine that we have exposed to many foreign antigens. Okay, so immediate action, rapid action in the intestine is very important, which is made by the presence of many activated effector cells. And controlling of these effector cells is very important on the other hand, which are controlled by regulatory cells, including M2, tolerogenic DC, and regulatory cells. Is that clear? So the first layer was epithelial layer, tight junction, mucus, IgA and antimicrobial peptide in the mucus, intraepithelial lymphocytes, and in the second layer we have had many cells that they are mainly activated effectors or regulatory cells. And if these two layers cannot successfully control the infection, we will have transferring of antigen to the nearby lymph node. Then, when we have antigen in the nearby lymph node, we will have a severe, a strong immune response by naive cells over there. Indeed, it's a danger. It's a signal for the nearby lymph nodes that something important is going to happen over there. So we need to take actions by induction of a strong immune response inside the nearby lymph nodes. The first layer, second layer, and the third layer. Clear or not? I hope it's clear for you. So you can see here the sampling of antigen by dendritic cells. Antigen in the lumen, epithelial cells, and they are sampled by dendritic cells. Or this is a sampling by M cell. And this one is transferring of IgA produced by plasma cells. So majority of plasma cells in the barriers produce antibody, which the class of antibody is
IgA.
The main role of IgA in the barriers is neutralization. Neutralization of what? Pathogens, any antigens. And mucus. So, when we talk about homeostasis, normal circumstances, we have just sampling and assessing or checking. And the main cells are tolerogenic DC, IL-10 or TGF-beta. We have it here. Regulatory T cells and B cells produce IgA. So when we have homeostasis or just microbes and we have normal flora, the situation is toward homeostasis by tolerogenic environment in the barrier. But when we have a pathogenic microorganisms, instead of tolerogenic DC, we have activated DC. Of course, there is M2 macrophages here, but here M1 macrophages. So, if there is a pathogenic microorganisms, instead of tolerogenic DC, we have activated DC. Instead of regulatory T cells, we have THR1 and THR17. Instead of anti-inflammatory cytokines, we have inflammatory cytokines here. Pro-inflammatory and inflammatory. We have, instead of M2, we have M1. This is M1. So, this is homeostasis, just sampling and assessing or checking of the content of the lumen by effector and regulatory cells. But in the inflammation, the story is different. So, unwanted immune response to normal flora of the intestine, I've told you that causes a disease, a group of diseases, which is called IBD, irritable or inflammatory bowel disease. Two types of those diseases is ulcerative colitis and Crohn's disease, that you will have at least one lecture about them in your clinical course. Unwanted immune response toward normal flora. In normal circumstances, in normal condition, we do not have immune response against normal flora. But if we induce immune response against normal flora, which is unwanted, we might have some disease. And another disease that we have, that there is unwanted immune response against gluten, dietary gluten, a food antigen, again unwanted immune response. So it's very, some people cannot keep the homeostasis in normal condition. There is a problem with their immune system, and there is no normal homeostasis. When there is no normal homeostasis, these three diseases at least have been identified in the intestine as an unwanted immune response. So, this is the last slide. Just a few seconds. No, not this one. Not this one. You know, we have many immune-related diseases of the skin. I want to cutaneous or skin immune system. We have many different diseases that you will have some lectures about some of them in your clinical course, including, for example, psoriasis, atopic dermatitis, urticaria, or, for example, different bullous diseases, pemphigus vulgaris, pemphigoid, bullous pemphigoid, and they are among the diseases that all of them are immune-related diseases of the skin. Indeed, in all of them, we have unwanted immune response in the skin. Some of them are autoimmunity, some of them are allergic reactions of the skin. So here again in the skin, we have three layers. The first layer is in the epithelial epidermis, and the second layer is in the basement membrane, and the third layer is draining lymph nodes. You can see here the epiderm and derm. The connective tissue of the skin is called derm, but the intestine connective tissue was called lamina propria. So again, we have some layers of epithelial cells, and inside the epithelial cells we have antimicrobial peptides, which are produced by some epithelial cells, We have intraepithelial lymphocytes, they are CD8 positive or gamma delta T cells. We have some specialized epidermal dendritic cells, which they are called Langerhans cells. They take the antigen bound by themselves to the nearby lymph node. And similar to intestine, we have different type of cells which they are spread in the connective tissue. The story is same, but the difference is the multi-layers of epithelial cells and presence of dendritic cells between the epithelial cells here, which they are called Langerhans cells. The rest of the cells and the rest of the story is as same as intestine. That's all. حفظكم الله. No question?